So hello, everyone, and welcome. We're glad to have you here today. As usual, if you wouldn't mind putting in the chat where you're viewing from, what fun birds are you seeing this week, and where did you see them?
People are coming in quickly.
Mhm.
Charlottesville, Virginia. Wow.
Sarasota. All right. Anybody seeing any good birds? We have folks from Sarasota and Lakewood Ranch, Wilmington, Delaware. Hello, Carol Brown. Okay, Lido Key, Black Crown Night Heron. Nice, Lynn Sheldon. We don't see those too often. Cynthia Escher in Bradenton, a lovely flock of roseate spoonbills at Mayaca River. Nice, River State Park. Maureen Dwyer, oh no, Ruth Marion from North Andover, Massachusetts. All right. Northern Shoveler, American Widgeon, and ringneck duck. Great. Maureen Dwyer, Haverford, Pennsylvania, where it's in the high 50s. Woohoo, nice. Carol Breedlove in Tacoma, Washington. See, I told you we get these great people from all over. A whooping swan, is that it? Nice. Jim Bensivenga from Venice. Two great sightings at Carlton Reserve last Wednesday at dusk. Bluebirds and a palm warbler was snatched out of the sky by a merlin and promptly eaten right by the log cabin near the entrance to Carlton. Wow, Nat Geo moment there, Jim. Great. Becky Griggs visiting us from Tulsa, Oklahoma. Welcome. Stan Orsi back in Maine. Stellar sea eagle 15 miles from their house. How's that for a backyard bird? Nice work, Stan and Joan.
I'm jealous.
Alan Gardner in Oakley Greens. He still has a daily robin and a greenback heron and some little blues. Nice.
Alan is my neighbor. Hi, Alan.
All right.
Jean speaking.
Kim Himmelfarb in Bradenton, seeing lots of black-bellied whistling ducks from the trail around the lake in Greenbrook in Lakewood Ranch. All right. All right, great. Thanks everyone for checking in. We love it. A few housekeeping items. Any questions you have, feel free to put in the chat, except for our speaker. If you have questions regarding her talk, you can pop them at any time into Q&A. So we would really appreciate you doing Q&A for any questions for the speaker. It allows us to keep track better. But if you do put them in the chat by accident, we will catch them there as well. And that's about it. We're going to give you a brief introduction here, and Jean's going to say a few things, and then Margie will introduce our speaker, Danielle Whittaker. So, nothing else new in the chat. I'll turn it over to Jean.
Have you shown all your screens, Karen?
Yeah, we'll go through the slides. There we go. Just a reminder to renew your membership, and you can do that online or drop it off at the Nature Center between 9 and noon. Also, the second Saturday just happened to be last Saturday, so our next garden day will be in March 11th. And we're getting some great turnout and lots of good info from our doctor of gardening, Pam Callender. And our next members only event will be in March with Catherine Clements from IFAS Extension in Sarasota. And she's a wonderful environmental educator and will bring us up to date on opportunities from IFAS, UF IFAS Extension. All right, I'm going to stop share and turn it over to Jean.
Hi, everybody. Am I on? Are we
Yes,
done? Okay.
You're on. You're on.
Okay. Thanks, everybody, for tuning in. Always wonderful that we see such a good crowd of people that log into our Zooms. Really wonderful. I can't wait to listen to our speaker today about the scent of birds, the sense of smell. It's going to be wonderful. But I have something very practical to talk about today. Within the next week or so, we will be dropping an annual appeal out to you. And you know that our overall programming is constantly expanding. This year, we did add our meet and greet afternoons at the Nature Center. And we now have our native plant expert who is happy to answer all your native plant questions on every second Saturday of the month. She's really wonderful, Pam Callender. Our wingspan game, there are many of you now involved in our wingspan game. It's managed by Lynn Jakubowicz and Martha Stroud. And the game has caught on and it's increasing every week. I think they have a couple of sessions every week. And I'm glad to say that our Celery Fields Explorers kids, that program is back in full swing after a 2-year hiatus because of COVID. So in order to keep all of these programs and everything else that we do going, we really need your support. And in order to keep the Nature Center going and the programming going. And I'll just tell you a couple of things that happened to us recently. We've encountered some unexpected expenses. For example, we have just had both fountains completely overhauled at a cost of close to $4,000. And the advice from the fountain experts is that we have to have the fountains maintained on a more regular basis. So we have to put that into our annual budget for next year, or for the remainder of this year and then next year. So we have to absorb these costs. And another unexpected cost is we have to add 50 feet of fencing to the back of our garden. And why is that? Because about a month ago, in the middle of the night, 12:30 a.m., somebody drove right down the hill and right back into our garden and damaged trees and signs, actually. And we estimate that the cost of the damage was about $1,000. But we now feel that we have to put the fence, extend the fence. There is one behind the bird feeding area, which we put in to stop people walking through there. And so we're going to extend this fence, and that's going to cost us about $1,500. So between those two things and the fountains, we have unexpected costs of $6,500 to $7,000. So they were not in our regular budget. So we are hoping that you will help us be able to defray those costs. And in the past, whenever we have asked you for help, you've always responded, and we really appreciate it. We heartily thank those of you who have donated. There's some of you who are constantly donating. And we heartily thank those of you who are going to donate as a result of this annual appeal. So thank you for supporting us. It's your Nature Center. It's not mine. It's not the board of directors. It's yours. It's ours. And we want to keep it going in the best way we can. And that's it. That's it for my appeal. And we're going to turn it over to Margie now, who is going to introduce our wonderful speaker.
So good evening, everybody. It's my pleasure. My real pleasure to introduce to you Danielle Whittaker, who will talk to us about avian scent, the secret perfume of birds. It's her research, and she's got a new book by similar name, The Secret Perfume of Birds, Uncovering the Science of Avian Scent. And Danielle, it looks like it's doing well if I check Amazon, which is always good to see how stats are going. So it sounds like people are really enjoying your book. It's the untold story of the stunning discovery. Not only can birds smell, but their scents can be the secret to their world. So Danielle is an evolutionary biologist and managing director of COLDEX Center for the oldest ice exploration, which is a National Science Foundation Science and Technology Center at Oregon State University in Corvallis, where she's been for slightly over a year. Before that, she was the managing director of another National Science Foundation funded Beacon Center for the study of evolution in action at Michigan State University for 12 years. Her primary research there was on birds, what they can smell, and what those smells tell us. Danielle has a PhD in biological anthropology from City University of New York. Yay, New York City. And on weekends, she's a roller derby referee for the Lansing Derby Vixens. Sounds like a hot bunch of ladies there. So it's my pleasure to introduce to you Danielle Whittaker. Take it away, please.
Thank you very much for that great introduction. I do regret to say I haven't done any roller derby since 2020, but it was a pretty awesome 10 years of my life, and some of that's in the book too. So I will go ahead and share my screen. All right, so please let me know if there's any problems. So thanks again for coming to hear me talk about my work. I am sorry to say I won't be talking about toucans, but I could not resist using this beautiful photograph because many of us associate the phrase "follow your nose," which I've titled my talk, with these birds because of the cartoon mascot of Fruit Loop cereal. And I actually find it a little what's the word? Ironic that we actually associate a bird with this phrase, because for a very long time there has been a myth that birds had little to no sense of smell. And that's where I would like to start today. So the chemical senses, which include our sense of smell and our sense of taste, have been called the most ancient and universal senses. And that's because they are found in all forms of life on this planet. They're very important for sensing your environment, sensing what's safe and what's not safe. And we can look at examples of this across all forms of life. So for example, bacteria display a behavior called chemotaxis, and this is a behavior they show in response to chemicals that they sense in their environment. If a chemical is beneficial to them, they will move towards it. That's where the taxis part of the word comes from. If it is harmful, they will move away. Plants also produce chemicals and communicate with them, and they even communicate with other species. So this picture here is of the tobacco plant. Tobacco plant is preyed upon by the tobacco hornworm, which is a kind of caterpillar that comes and chews on the leaves. Obviously, this is not ideal for the plant. And so they have evolved this system where when the tobacco hornworm chews on the leaves, its saliva mixes with another chemical in the plant's leaves that produces an airborne compound that is actually sensed by yet another species of insect, the big-eyed bug, which is attracted, and it comes and it eats the tobacco hornworm, which I think is a really amazing example of interspecies communication and cooperation. Thinking more about animals, you're probably familiar with what salmon do in the breeding season, where they all swim upstream to the spawning grounds, and you'll see hundreds of them all at once, and that's because they're following a pheromone in the water. So when they're in breeding condition, salmon will release this chemical compound from their skin into the water. It's called an aggregation pheromone. So when you see lots of animals, the same kind of animal together, in this case, the salmon, they're all releasing the same compounds. So another salmon in the river will sense where that compound is coming from, because the more salmon that are releasing it, the stronger the signal. And that's how they all find each other and find where to go. And then even closer to home, you're probably all quite familiar with the idea that dogs have a good sense of smell, in fact, an exceptional sense of smell. They're known to be able to find their way home over long distances, for example, to detect contraband in your suitcase at the airport. And if they're trained to do so, they can even detect cancer through smell. So these senses are very important for interacting with the world around us. They're found across all forms of life, which is makes what I'm about to say next really strange. And that's that we still have this idea that's been around for a while that birds aren't able to do this. This is the Wikipedia entry for olfaction, the sense of smell. And the first sentence, well, one of the first sentences reads, "The importance and sensitivity of smell varies among different organisms. Most mammals have a good sense of smell, whereas most birds do not." And then it lists a few widely accepted exceptions to this so-called rule. The first thing I want to point out is there's no citation to this statement, and that's because there's actually no scientific evidence that this is true. When I first learned about this, I was a postdoc at Indiana University. I had just joined a lab studying birds. I didn't have a lot of background in ornithology, and a fellow scientist said to me that birds didn't have a sense of smell, which I thought was the strangest thing I'd ever heard. And I tried to track down the source of that statement, and it was pretty tough to find. And so this became a really interesting topic to me. And I did eventually discover the source of the idea and why it persisted. And so now I'm going to give you just a very brief history of the study of avian olfaction. I could talk for a long time about this, but I'll just give you the highlights right now. So this idea actually dates back to John James Audubon himself. He published a paper in 1826 with the fabulous title, "Account of the Habits of the Turkey Buzzard, particularly with the view of exploding the opinion generally entertained of its extraordinary power of smelling." So I love titles from this era because they all tell a story like this one does, right? So clearly people already believed that turkey vultures, turkey buzzards, same thing, were able to detect the scent of a dead animal. So like now we see, a vulture circling overhead and we think, oh, there must be, roadkill out there. And then that's because they're detecting it by scent. Audubon didn't believe this could possibly be true. He knew that birds had extraordinary vision, and in some cases relied very heavily on their auditory senses. So many birds are known for song. And he believed that, and this was not an uncommon idea, he wasn't crazy for believing this. He believed that if an animal had evolved one sense so well, so in this case vision, they must have had to give something else up. And why not smell, he thought. And he wasn't the only one who thought this. And so he was clearly annoyed at this belief that they used their smell to find food. And he did a series of actually very poorly planned experiments, which he believed showed that they did not use smell, but instead used sight. So an example of one of those experiments was he took a rotting pig, put it under the bushes, and waited for the vultures to come. And when they didn't, he said, "See, it's because they couldn't see the pig. They didn't know where it was, and therefore they couldn't find it." However, if you actually read the paper, he does also talk about how the dogs were there all day attacking the dead pig, and there were vultures circling around overhead, which I think shows the opposite of what he's claiming it shows. So that paper set off several decades, actually, of discussion and some amateur ornithology research. A lot of different people put a lot of time and effort into trying to either prove or disprove the idea that birds could or could not smell. And it wasn't really until the mid-1900s where we started getting well-designed research aimed at understanding the sense of smell of birds. And I'm going to start with Kenneth Steger because his subject of interest was the turkey vulture. And he did his PhD thesis on this, and then became the curator of birds at the University of Southern California Museum of Natural History. And he was interested in the same question, but luckily for him, by chance, he met an engineer from the Union Oil Company who happened to tell him that the engineers from the company would use vultures to help find leaks in natural gas lines. So if you've ever smelled natural gas, I'm sure most of us have, it does not smell very good. Well, actually, natural gas doesn't have any smell at all. It's completely odorless. And they add a chemical called ethyl mercaptan to the gas, because otherwise, if there's a leak, it can lead to catastrophic explosions because we'll never sense it. So this ethyl mercaptan, it smells very bad, and it just so happens to be the exact same chemical that's given off by dead animals. And that is what turkey vultures are attracted to. And that's what you see them finding when they're circling over the area where there's a dead animal. So if there's a natural gas leak for a very long time, these engineers have known to look for the vultures to help localize where that leak is located. And so Kenneth Steger used this information to test for sure that that's what they were doing. And he created a little device that gave off this ethyl mercaptan and studied how the birds reacted to it. And he found very clearly that, yes, they were detecting the scent of this, and that's what they were honing in on. And in fact, it worked so well that whenever he used the device, the vultures would be circling overhead for just hours and hours until he ran out of ethyl mercaptan. So it was very clear. So this research continued around this time with a few big names in this small field, including Bernice Wenzel, who was a pioneering biologist studying the sense of smell in a number of different bird species, including many seabirds like this albatross. So seabirds are widely accepted as an exception to the so-called rule for a number of reasons. So Procellariiformes is the order that includes all these tube-nosed seabirds. They're called tube-nosed because many of them, you also can see on the outside of their beaks this tube-like projection, which is actually their nostrils, basically. And so it's clear that they've got some anatomy dedicated to smell. And so people seem to readily accept the idea that these birds have a sense of smell. And in fact, there's been some really cool research showing how they use smell to find food when they're out hunting over miles and miles of open ocean. So they're out there looking for food like fish and squid. But what it turns out that they're attracted to is a chemical called dimethyl sulfide, which is given off not by the fish or the squid, but by the phytoplankton. So when there's a large group of phytoplankton in the water, they will give off this odor that comes up off of the water. And where there's a lot of phytoplankton, there's a lot of fish and a lot of other animals all feeding. And so that's where they go to find their food. So again, this work was originally led in the 60s by Bernice Wenzel, picked up later by Gabrielle Nevitt, who's at UC Davis. And this group of birds, again, everybody's like, yeah, sure, these birds smell. This is where we first started seeing studies looking at whether birds use odor in social behavior. So one of these first studies was on the crested auklet, which are these cute little guys. These are Arctic seabirds. And in the breeding season, when they're in breeding condition, they give off a very strong tangerine-like odor. And when these guys meet each other, they walk up to each other and they do this behavior, which you can see in this picture here, called a rough sniff. So they will go up to each other and put their beaks right in the feathers at the nape of the neck, which happens to be where this odor is strongest on the bird's body. And they'll get a good sniff of what this odor is like there. And so Julie Hagelin and her advisor, Ian Jones, before her, did some important first work on this, learning about the role of this smell in their social behavior. It seemed to be a very important part of their interactions. And then Francesco Bonadonna, a French researcher, started studying petrels, many different species of petrels, which are small seabirds, which are found in the southern oceans. And these guys, they nest in colonial colonies with hundreds and hundreds of birds there, and they actually nest in burrows, so underground. And on top of that, they are out foraging all day, and they tend to come back to their nest at night. So they have to find their burrow in the dark, surrounded by all these other burrows of other birds. And he believed that they did that through smell. And he tested this and found that was correct. That was actually how they were doing it. So this is a drawing from the original paper. You can see this box in the middle. This is where they would capture the petrel, and they would put two tubes from the box. And one was to that petrel's own burrow, and one was to a different burrow. And they found that with nothing out to guide them other than their sense of smell, they were able to choose the correct burrow. So pretty good evidence that they were using smell. So I've been talking a lot about seabirds, vultures, but what about songbirds, right? The passeriformes, or the songbirds, make up over half of all species of birds. It's over 6,000 species. These are the birds that many of us encounter most often in our daily lives. Many of them are brightly colored or have elaborate songs. So they are known for having, a very important role of vision and hearing in their daily life. But their sense of smell was generally very much dismissed. So they don't have the big beaks like the seabirds, and their brains are on the small side. And in fact, when I was first told that birds had no sense of smell, it was by an avian neurobiologist who was telling me, well, their olfactory bulb, the part of the brain that receives signals from your olfactory receptors in your nose, so that's where you're learning about the smell around you. It's so small that they didn't think it was functional. And in fact, when he dissected brains for his research, he would actually throw away the olfactory bulb because it was useless. Well, I was told this as a young postdoc who had just started work in a bird lab, and I thought, well, that's the silliest thing I've ever heard. How could this huge group of animals, so many different species, be lacking the sense that we know is so important for survival because it helps you sense the world around you, find food, avoid poison, all that kind of stuff. So we do know, and we were starting to learn at that time, that songbirds do produce odors like every other animal. And that's primarily from preen oil secreted from the uropygial gland, which is located above the base of the tail. This crossbill is showing us where his is. It's called preen oil because while birds are preening, they will reach back and take oil with their bills from this gland, and they'll rub it on their feathers while they're maintaining their feathers. It helps protect the feathers from exposure to the environment. It helps protect the birds from ectoparasites. It helps with thermoregulation. It helps with waterproofing. And it also gives off airborne or volatile compounds that give the bird its smell. And before I go any further, I just want to introduce you to the method that we use to study smell. So it's, as you might imagine, it can be a little bit challenging because you can't see it, you can't hold it, you can't touch it. It's the air, basically. So fortunately, there are methods developed by very smart chemists to do this, and we use a method called gas chromatography mass spectrometry, or GCMS. So chromatography is a method that you use to separate out all of the compounds in a mixture. So you can do this with liquid, you can do it with gas. Here we're doing it with gases, obviously. And it sorts them on the basis of their size. And so mass spectrometry is a method where you use lasers to detect what those compounds are, what those molecules are, and you can get information about their molecular mass and their size, and you can compare it against a database to identify what those compounds actually are. So here on the screen, I've got two GCMS results. In the middle there, on the top is GCMS results from the preen oil of a gray catbird, who's pictured on the left. And on the bottom is the preen oil from a northern cardinal, pictured on the right. And you'll see on the bottom line, the x-axis, we've got some numbers here. This is time in minutes. So what is happening here is that as you force the gas through this tiny tube in the instrument, the smaller molecules will travel through more quickly, and the larger molecules will take longer to go through. So that's what you're seeing here. Smaller molecules come off first, so 5 minutes, 10 minutes, and so on, and then larger molecules further on. And then, of course, we're taking a measure of the abundance of those molecules, so the intensity that the machine is seeing, and that's this y-axis. And then we could take this information and then identify the compounds and then measure how much of them are present in this gas. So you might notice there's some differences between these two species. There's also some similarities in what compounds might be there. I see greater diversity in the gray catbird preen oil, for example, more compounds. And what we do tend to see is that each species has its own species-specific set of compounds that make up a species-specific odor. But individuals within that species will vary. So they'll have their own personal odor within those parameters. So next I'll introduce you to my study species. You're likely already familiar with them. The dark-eyed junco is a widespread North American sparrow, which breeds in high latitudes, like up in Canada, and high altitudes in the Appalachian and Rocky Mountains. They are also often seen in backyards at bird feeders in the wintertime, so in many places they're also called snowbirds. I joined a lab. I joined Dr. Ellen Ketterson's lab at Indiana University in 2006 as a postdoc, which is where I started my journey studying birds. And she has maintained a long-term study of juncos at Mountain Lake Biological Station in Virginia, which is located here. She's been working with them since the 80s, and that study is still going on today. So it was a great opportunity for me to get involved and work on this species. Juncos are really a great model species to use if you're interested in songbird behavior, because they're like a lot of other birds in their behavior. They're pretty normal little guys. They're also interesting because in the last glaciation, about 10,000 years ago, they diverged into several different subspecies, and you can see some of those here. I've got some photographs of some of the ones I've been lucky enough to work with. I've worked primarily with the slate-colored junco on the East Coast, so mostly at Mountain Lake Biological Station, and also Oregon juncos in San Diego County in California. But now that I live in Oregon, I'm lucky enough to have them in my backyard right now. So that was where I was first told that birds had no sense of smell. That's where we were starting to think about preen oil. And another member of the lab was really interested in this idea of preen oil and wondering, the role it might play in hiding the nest from predators. So juncos are ground nesters. This is a junco nest. This is just on the dirt underneath a little bit of foliage. And predators find their nests all the time. So their eggs will get eaten by snakes, by raptors, by rodents. The chipmunks are the worst, at least at Mountain Lake. And around the same time, a study came out on shorebirds, especially these red knots here, looking at their preen waxes. They call them waxes in birds like shorebirds because the waterproofing is so much more important, that it's a lot waxier for them. And they found that they were changing the composition of their preen waxes when they were nesting. And in fact, this researcher used a dog that was trained to find the red knot nests. And it turns out, so they're switching from what's called monoester preen waxes into diester preen waxes. And the chemistry is not that important right now, but what is important is that the diester preen waxes are harder for predators to smell. And with the dog that was trained to find the nest, that is exactly what they found. When they switched their preen waxes, the dog couldn't find the nest as easily. So my colleague in the Ketterson lab said, well, we would expect then that the preen oil of juncos would be less smelly during the breeding season to camouflage the nest from predators, just like you see in the shorebirds. The results, however, show the exact opposite. So here we have, along the bottom are a set of linear alcohols that we always see in junco preen oil. And then on the y-axis is the concentration. And then we've got green bars are from birds in breeding condition, and the dark blue bars are the results from birds in non-breeding condition. And right away, you should see what we saw, and that is that they produce way more of these compounds when they're in breeding condition. So I saw this and I said, oh, wow, this must be important in breeding. This must be important maybe in mate choice, mate attraction. And I kind of took this idea and ran with it. So for the rest of the talk, I'll talk a bit about what I've found in my research, and I'll include some research from other people as well, because this area has really grown since I began studying it. A lot of people have become interested, and there's been some really great work. So I'm going to talk primarily about how songbirds use odor in social contexts during the breeding season. So I'll be focusing on mating, on fighting with potential rivals, and on raising offspring. So first, let's talk about how birds use odor to find mates. So again, the first thing I thought of when I saw these results that I just showed on the previous slide is that, wow, maybe this is important for attracting mates. Maybe it's not just songs, maybe it's not just flashy plumage, maybe there's some kind of smell that's attracting them. And the first thing that comes to mind when you start talking about something like that is the word pheromones. And we're not talking about pheromones here. So pheromone is a very specific molecule that, sorry, my dog is barking. I hope you can't hear him. That has that elicits a very specific response in the receiver of that molecule. So here in this drawing is a diagram of moth pheromones. So moths are one of the best known examples of this. Females will give off a pheromone in the breeding season, and males are attracted to it. And as soon as they detect it, they will go right towards it. It's a very specific response. Another thing about pheromones is all female moths in the same species all give off the exact same molecule, right? It doesn't give that male any information about that female. It doesn't say who she is, what kind of health she's in. It just says, oh, there's a female who's looking to mate. I better go to her. What we're looking at when we're talking about these more complex odors, like what we're seeing in preen oil, is what's better known as a signature mixture. So we're going to see variation among individuals. Like they all have the same compounds, but their individual mixture will be a little bit different. And these mixtures provide information about that animal's biology and their identity and their quality. So in a way, it's less like a pheromone and more of an analog to, say, birdsong, right? So, you know, all the males in the same species, let's say cardinals, right? We know, many of us know what cardinal song sounds like, but we also know that there's variation in what individual males might sound like. So the females know that's a male cardinal, and then they can listen to him and decide whether he sounds good or not, right? So that's more what we're thinking of with these odors. So to answer the question of whether birds use odor to attract mates, I used what's known as an odor preference test. So here I constructed a plexiglass Y maze. I hope you can see why it's called a Y maze. That's how it's shaped. And what you do is you take whatever you're asking the animals to choose between. In this case, I used cotton balls with a bit of preen oil on each one. You put one on each end of the Y maze. You put your animal in the beginning here, and then you leave the room so you don't bother them and let them spend some time and decide which arm of the Y they want to spend more time in. And you videotape the results. So here we've got Kate, who was my undergraduate assistant at the time. She's putting preen oil on those cotton balls there. And then here is a still from one of the videos. You can just make out the junco here hanging out in arm number 2, because that bird seems to prefer that odor. So I asked, is this an attractive odor? Is it important in attracting mates? And what I expected to find, if that were true, would be that the males would prefer to go towards the female preen oil odor, and that females would prefer to spend time with male preen oil. It's not exactly what I found. So here we've got our subjects. I had 22 males, 18 females, and then these are just bar graphs showing the average number of seconds they spent in each arm of the maze, whether it was with the male preen oil in blue or female preen oil in pink. And you can see that males overwhelmingly preferred the scent of other males. Well, whether or not they preferred it is maybe questionable. That's where they spent the most time. Females seem to go half and half. There's no clear preference here. So clearly this is not a mate attraction odor. But I also want to give the caveat here that a lot of times, this will come up a lot when you're looking at studies of mate choice behavior in birds and other species. When you put them in these artificial situations and ask them to choose between something, it's not really replicating actual mate choice, right? They weren't smelling 2 actual birds. They weren't looking for a mate. They were in a plexiglass cage in a laboratory. So who knows what they were thinking, right? So we need to take those kinds of studies with a grain of salt. But I started thinking, okay, well, maybe it's not for mate attraction, but it could be for mate assessment. And I'm not going to go through every result we had, but I have found that the information present in the preen oil does correlate with aspects of the bird's biology and can be useful when, let's say, a female is deciding between 2 different males, or she's looking at a male she just met. There's information available to her. And one of the most intriguing things that she can learn about is a particular gene or gene family, I should say, the major histocompatibility complex, or MHC. This is a very large family of immune genes. So we have these as well. All vertebrates have them. And these genes are involved in your body's ability to detect and fight off pathogens. And so there's been a lot of studies looking at how this actually might be important in mate choice in a lot of different species, including humans. So the idea here is you don't have just one MHC gene, you actually have a lot, dozens, and you don't have the exact same copy at each of those dozen sites in your genome. If you did, Just because you had a dozen of them would not help you. That would mean that you would only be able to detect and respond to that one kind of pathogen that that particular gene codes it for. Instead, what you want, ideally, is to have a lot of different copies of this gene at all of those sites in your genome. And that increases your ability to detect a variety of pathogens and mount your body's immune response to them. So the idea here is that if you're looking for a mate and you want to make sure that your offspring will be healthy and be able to fight off diseases, the best choice would be another member of your species with different MHC genes from your own. And that way, when you mate, you're giving two different sets, or half of each of yours. You're giving a mixture of different copies of this gene, and your offspring will have a nice, good, healthy immune system. There's been evidence for this in a lot of different species, including humans, although it can be hard to interpret sometimes. But this has been studied in preen oil recently in song sparrows. So this is research coming out of the McDougall-Shackleton lab at Western University in London, Ontario. Joel Slade, who was my postdoc for a while, but during his PhD, he studied this question, and he found that song sparrow preen oil odor did give information about MHC genotype. And specifically, if you take two song sparrows and you measure the chemical distance between their odors, so there's a way to calculate that, so how different their odors are, it correlates with how different their MHC genes are. So, okay, if you're looking for someone different, you need to find someone with a more different smell than yours, right? And then a few years later, Leanne Greaves in the same lab did some behavioral studies and found that indeed, when given a choice in a Y maze, much like the one I used, the song sparrows prefer the scent of an opposite sex bird with MHC dissimilar genes. So that supports this hypothesis that this could be important in mate assessment. They're more attracted to that scent. But I've been kind of focusing on the idea of females choosing between males. That's often what we think of, especially in birds, especially because males tend to be the sex that sings and males tend to be the birds with the brighter colored feathers. But in fact, females are not just passive little choosers. They are also making some advertisements themselves. So let's think a little bit about female chemical signals. Those results I showed you with a higher concentration in the breeding season than the non-breeding season were from males. But we also found that females show the same pattern. And importantly, females actually produce more of these compounds than males do. So one of the first breeding seasons in the wild that I studied this question, I collected preen oil from all of the juncos in our study area at the beginning of the breeding season, so like April. And this is the time when they are finding mates, when they are staking out territories. The males are singing, the females are building nests. They're starting to lay eggs. There's a lot of hormonal changes happening here, right? This is a very interesting time biologically. And so I measured their preen oil odor across that month, basically. And in males, we would see just a steady incline of these volatile compounds over those first 4 weeks. But in females, the pattern is different. So here we've got, these are some methyl ketones, which tend to be higher in females than males. And this is over those first 4 weeks of the breeding season. And you can see there's no difference between weeks 1 and 2 in abundance. But then there's a huge jump in week 3, and then it levels off a little bit in week 4. And I wasn't sure what to make of these results at first. And I thought, well, what was different about week 3? What was going on in the population at that time? So I looked back at all of our behavior logs, all of the, the study site data from that season. And I found that week 3 was when most of the females were laying eggs. So this is when they're ovulating. So they tend to mate only 1 or 2 days before laying that first egg. So that would be the same week. This is when it's most important for them to mate with a male so they can fertilize those eggs. And this is when you would expect them to be advertising that, hey, I'm ready to mate now. And that way they can get the timing correct. And this would also correlate with changes in hormones. They tend to be highest in testosterone at this time, for example, as well as estradiol and progesterone. So this could actually be very important for female behavior in a way that we hadn't really thought about much yet. I will say there was another interesting study on chickens. showing again that female chemical signals might be really important for mating. And so here we've got a study where they gave males the opportunity to mate with females. And half of the females had their uropygial glands, perfectly normal. And the other half of the females had their uropygial glands surgically removed. And they were otherwise the same, behaving the same. And so the males were given the opportunity, and they found that males were far more likely to attempt to breed with the females who had their preen glands. So these are the birds, the white bars here, then compared to when they were given females who had their glands removed, which are the black bars. And so this suggests that it's possible that the chemical signals or the odors from the preen oil might be what stimulates the male to want to mate in the first place. It could increases testosterone production, for example, and so it could be very important, again, in the timing of mating. Okay, so I've been talking a lot about mating, but that's not the only thing that birds do, not even in the breeding season. So for example, there's a lot of competition. It's competition for territories, competition for food, competition for mates. And so this is more thinking about within sex interactions. So males tend to compete with males, females tend to compete with females. And we're learning that odor can be important there as well. And so I want to start with this study. This is another study that was designed to be like a mate choice trial, similar to what I originally did. But again, as I noted earlier, you're not always measuring what you think you're measuring. So this is a study by Louisa Amo, who was studying house finches. And they did a very similar setup to what we had done. So the idea is that you put a bird in the middle and they have to make a choice between one side or the other. They improved upon the study in one very important way, though. They actually used whole birds in the ends of this apparatus. So not just the odor, but the whole birds. And these birds, so here on either end, they're in a darkened chamber in a cage. So when songbirds are in the dark, they usually don't move around much. They don't sing. So this male in the middle, who's having to decide which way he wants to go, he can't hear or see these birds. He can only smell them, but it is a whole bird body odor that he's smelling. And so they had a male in the middle, and they gave him a female on one side, a male on the other side. And they said, well, if this is important in mate choice or mate attraction, we would expect him to choose the female. Well, that's not exactly what happened. So about half of the birds chose the female, 56%, and a little under half of the birds went to the male side. I love this paper because they went back to try to figure out what's going on, and they found out something really interesting. So what they did was they went back and they measured the quality of that male in the middle, and then the males, what we call the donor males, or donating their odor, and compared them. So the quality score was based on a couple of different things, including a measurement of body condition, an immune response measurement, and their feather coloration. So the brighter colored a house finch is, it tends to be in better health. And what they found is that when that focal male, the guy in the middle here, when he was in better condition than the donor male, the one in this darkened cage, he would go towards that male side. But if he was in worse condition than the donor male, then he would avoid him and go to the side with the female. So it's not necessarily that he was attracted to the scent of the female. Instead, it seemed that he was avoiding a male that might be able to beat him up. So this wasn't exactly what they had expected to test, but it was, I think, the first example we had of how odor might be important in this kind of rivalry, assessing a rival and deciding whether or not to fight. So a few years later, I teamed up with my colleague, Kim Rosval, who's done a lot of research on aggression in birds, in males and females. And we decided to study this question in juncos. So we collected preen oil from wild juncos, and then we did a behavioral trial, and it's called a simulated territorial intrusion. And the way this works is you take a caged bird and you place it in the middle of a territory of a wild bird. And next to that cage, you have a little speaker and you play Junko song. And this will enrage the male whose territory you're on, and sometimes the female too. And they will come out and behave very aggressively towards that bird in the cage. One of those behaviors is called a flyover. They're just swooping very closely and aggressively over the cage. And there's other aggressive behaviors as well that all tend to correlate with each other. And what we found is that the odor, and this is a composite measurement of different aspects of the odor, correlates significantly with their aggressive response. And so in males, you can see the higher their score for the odor, the more flyovers they did over the cage. In females, there's also a significant relationship. It goes in the other direction, which is interesting, but it does tell us that there is information about their aggressiveness in their odor. And we also found that in males, at least, these odor profiles correlated with their circulating testosterone levels in their blood, and also with the level of androgen receptor expression in the uropygial gland. So androgen receptors, they detect testosterone and other androgens in the bloodstream. We have different kinds of receptors all over our bodies and all of our tissues. And that is how our body communicates with itself. So when we have certain hormones that will turn on certain functions in different tissues, it's those receptors that are sensing the hormones. So the more receptors you have, the more sensitive that tissue is to that hormone. So here what we're seeing is males that were very aggressive, males that had more aggressive smell also had higher testosterone levels, and their uropygial glands were more sensitive to testosterone, which shows us a pathway through which that information can correlate. So the testosterone is related to their aggression level, also related to their smell. Okay, and then the third type of behavior I want to talk about is recognizing relatives, which can be really important in a couple of different ways in a bird's life. So first of all, we know that parents and nestlings can recognize each other by smell alone, and this is true in mammals as well. Remember, I showed you the petrels near the beginning of the talk, those birds that burrow in the dark and are able to recognize the scent of their burrow just on the basis of their smell. Their nestlings can do that as well. So here is a petrel with its very fluffy chick. So yeah, there were more studies looking at that. This is true of zebra finches as well. There's been some good studies out of Barbara Kasper's lab in Bielefeld, Germany, looking at the odor recognition. So the parents recognize the scent of their nest, the nestlings recognize the scent of their nest, and the nestlings recognize the scent of their parents. So if you take a nestling, a brand new nestling zebra finch, and you give it the odor of its mother and the odor of an unrelated female, you can also do this with its father and the odor of an unrelated male, it will beg more earnestly towards the scent of its genetic parent. What's really fascinating about this is that is true even if you cross-fostered these chicks when they were still in eggs. So cross-fostering is a method where, say, you've got two nests that are at the same stage, they're about the same age, they were laid on the same day, and you can take those birds and switch them between the nests so that they are raised by different parents. And that helps you understand the difference between nature and nurture. Basically, it's a nice way to separate out genetic versus environmental influences on birds' development. Well, if you do this with these zebra finches before they even hatch out of the egg, they can still recognize the scent of their genetic parents, which is fascinating. And the reason for that is because they start learning scents while they're still in the egg. And so we know this from a study by Julie Hagelin, who studied this in chickens, and she presented embryonic chicks while they were still in the egg with some odors that weren't normal for them. And I think it was either citrus odor or one of those kinds of odors that they wouldn't normally encounter while in an egg. And then after they hatched, she did a standard, it's called a habituation-dishabituation protocol, to test whether they found that odor familiar and if they, compared to a completely different odor. And she found that, yes, they did learn those odors while they were still in the egg, so that the eggshell is permeable and their sense of smell is working already. So this is a very helpful thing. It's very important for birds and actually all animals to be able to recognize kin. When you think about how birds reproduce, they have many birds will just have 1 or 2 clutches per breeding season, per year, and they'll have subsequent years where they continue to breed, but those siblings will never have met their siblings from previous years, right? Because the older birds have already left home. But all of these birds grow up to be adults and have to go out and find mates. And it's very advantageous to avoid mating with your kin because inbreeding can be problematic, especially in populations with low genetic diversity. It can increase the likelihood that detrimental recessive alleles will be expressed. And so it's kind of a universal that animals avoid mating with relatives, with close relatives. And so back to our petrels, Bonadonna did a study with European storm petrels. This is another Y-maze experiment, presented them with odors of adults, either a close relative that they had never met or an unrelated bird. These would be opposite sex birds. And they overwhelmingly preferred the scent of the non-relative. So they were avoiding the relatives. And this is true in zebra finches as well. This is again the Casper's Lab. They did this study to test whether females would avoid mating with an unfamiliar brother. And they did this by placing the birds housing them in triplets. So instead of pairs, it would be 1 female and 2 males. One of those males would be unrelated to the female, and the other would be a brother, an unfamiliar brother, so from a different year. And they expected that if the female's sense of smell was working, then she would not ever breed with the brother. Instead, all of the females in that group, in that condition, would mate with the unrelated male. For comparison, they had half of the birds in the study, the females had their sense of smell blocked. They gave a zinc wash to their nasal passages, which temporarily blocks the functioning of the olfactory receptors. So those birds were functionally anosmic, which means they have no sense of smell. And they expected that those females would mate randomly with either the related bird or the unrelated bird. This is yet another study where things didn't quite go according to plan. But here we have graphs showing those 2 sets of females and the stage of breeding that they made it to. So of course, they all start out at 100%, and then the line goes down. So here on the top in the red are the females who do not have a sense of smell. So you can see, most of them made it to egg laying, fewer made it to hatching, but most of those did fledge their nestlings. And then on the black line were the females who could smell, and almost none of them reproduced at all. Like they dropped off at every stage here. So at first glance, well, okay, I guess that supports the hypothesis, right? The females who were housed with brothers and could smell them avoided breeding altogether. But one of the things that happened here was that those females who had their sense of smell became very aggressive, and they attacked the males, and to the point where many of the birds had to be removed from the experiment. And they didn't actually get to see the aggressive behavior because it happened at night, but they think what was going on was that those females felt threatened by having to be in a cage with a close relative that they didn't want to mate with. And so they responded by behaving aggressively and avoiding breeding altogether. So again, smell clearly very important here because that did not happen in the cages with the females who could not smell. So I was also interested in the question of this familial odor. We see this in all species. Family members do smell alike. And I had been getting questions about, well, how do birds produce these odors? What's the genetic basis of it? And around the same time, I met a microbiologist who studied mammalian odor. And it turns out that most odors in mammals, including us, are actually produced by bacteria, symbiotic bacteria that live on the body. In many mammals who have like scent pouches or scent glands, those are full of bacteria that feed on the secretions of the body. And those bacteria give off the odors that we think of as the animal smell. So this was Kevin Tice, my colleague, and I teamed up to look at this in birds. It had never been studied in birds at that point. But we hypothesized that uropygial glands would also be a great place to grow bacteria, and perhaps they were the source of this odor. So in order to look at this, I went out and I sampled 9 families of juncos. This is again at Mountain Lake Biological Station in Virginia. So this is a very crude map that I drew of our study site. So Mountain Lake, this is a mountaintop lake, which is pretty unusual. It's what the area is named for. There's a hotel right next to it. If you've ever seen the movie Dirty Dancing, you have seen this hotel. That is where it was filmed. This black line here, this is the road. All this is roads. And the road between the hotel and Mountain Lake Biological Station up here is about 2 miles. And this whole area is surrounded by forest. And that's where Ellen Ketterson's long-term study of dark-eyed juncos is located. And that's where I worked as a postdoc. And that's where I conducted the study. So here on the map, I have marked out all of the nests that I sampled. They're color-coded, which will be important on the next slide. And I also did paternity tests because, as it turns out, like many songbirds and other species, to be frank, they are not always genetically monogamous, even though they are socially monogamous. And so in every population of juncos that we've studied so far, about 25 to 30% of the nestlings were sired by an extra pair male, meaning that the male that is helping raise them is not their real father. So in this study, I did paternity tests, and I found 3 of my 9 nests had only within pair young. So the females were monogamous, only mated with that male. 2 of the nest had only extra pair young. So they were not fathered at all by the male who was raising them. And the remaining 4 nests had mixed paternity. So some of the nestlings were fathered by their social fathers and some were not. And so that would allow me to compare relatedness, the importance of relatedness and smelling alike. So first I looked at the bacteria. We used a genetic method to characterize the bacterial communities. So you can use DNA to identify what bacteria are there, and then you can do some measurements on the whole community. So we found that uropygial glands are indeed home to very rich and diverse bacterial communities. And that family members had very similar bacterial communities. So here, this is a multivariate analysis. Details aren't super important here. It's using something called Bray-Curtis similarity. So the closer the dots are to each other, the more similar they are. So here we've got the adults are in the the colored in circles. And the nestlings are the open circles, and the colors correspond to which nest they came from. So the first thing you might notice is that adults and nestlings are different. And that is indeed what we saw. There is an age difference here. But then within those age classes, you should notice that the different colors are clustering together. So down at the bottom here, we've got the 3 purple nestlings, all very similar to each other. Their purple parents are up here close to each other, and dark blue nestlings here and their parents there. So yes, what we're seeing is members of the same nest have similar bacterial communities. And I did a similar analysis with odor. This one's a little harder to see because I've got this outlier here, but the results are essentially the same. Adults and nestlings are a little bit different, but then within age class, we see members of the same nest clustering together. So you're from the same nest, you have the same bacteria, and you smell the same. Then if we look at within families, it gets even more interesting, I think. So here again, I'm using the Bray-Curtis similarity index. It goes from 0 to 1. The closer to 1, the more similar the 2 things that you're comparing are. Closer to 0, the more dissimilar they are. So here I'm comparing parent-offspring pairs, and I'm showing you their average similarity. The first thing we noticed is that mother-offspring pairs were more similar to each other at their bacteria than father-offspring pairs. This makes perfect sense. Only females sit on the nest. Only females incubate the eggs or brood the nestlings. They're on top of them all the time. Of course, they're sharing bacteria. Fathers, of course, do interact with their nestlings in this species. They help feed them. They do have physical interaction. So we do expect to see some similarity here. The next interesting thing is that it didn't matter if the father raising them was related or not. So in the cases where it was their social father, but not their genetic father, he was just as similar to them as the cases where the social father was their actual biological father. And then finally, we looked at siblings, because remember, some of my nests had multiple fathers. And so we could compare full siblings versus mixed paternity with half siblings, and we found no difference in the level of bacterial sharing there. You'll also notice that the siblings are more similar to each other than they were to their parents. So again, think about a nest. A junco nest is not much bigger than my hand, and you've got 3 or 4, sometimes even 5 eggs in there that grow from a small egg to basically an adult-sized bird in 12 days. So it's pretty tight quarters by the end there, and they're sharing tons of bacteria, right? So this all makes intuitive sense. And it's really interesting. We did more work on bacteria after this. So again, as I mentioned, we found the uropygial glands are home to a diverse community of bacteria, which we have since discovered are odor-producing bacteria, as hypothesized. We did a study where we injected antibiotics directly into the uropygial gland of captive birds and measured the effect on their preen oil. And we found that they produce significantly lower amounts of those compounds, the volatile compounds. And then we followed that study up by cultivating the bacteria. Excuse me. This photograph here on the top right is a scanning electron microscope photograph I took of preen oil, and some of these round blobs here are bacteria. But then I worked with a microbiology grad student, Jonathan Greenberg here, who took preen oil samples and put them on Petri dishes and cultivated the bacteria on those dishes. And then we measured the odor given off by those bacteria without a bird in sight, right? And they produced the same odors that we were measuring in the juncos. So we got pretty solid evidence that these odors are produced by symbiotic bacteria and not by the bird's own biological processes. So I'm going to sum up now. I've given you a lot of information here, but we have discovered that songbird preen oil odor is important. It exists, they can detect it, and it can be important in a number of behaviors, including mate assessment. So it gives information about individual quality. And one of the things I pointed out was MHC genotype, so those immune genes that could be important in attracting a mate. And even more than that, they seem to be really important for females to advertise their reproductive readiness. So remember, it correlated, the timing of the increase correlated with when the female was ovulating. And that could be important in stimulating males to want to mate. Could also be very important in competition. So it can help you assess a rival's likelihood of aggression in response to an intruder. And it's important in recognizing relatives, especially those you've never met before, and avoiding mating with them. And we found that preen oil odors are produced by symbiotic bacteria, but they're also influenced by the bird's own biology, because your bacteria are also influenced by your hormones, right? So we see these correlations with the bird's own hormones, and that's because those hormones also affect your bacteria, and bacteria can even feed on hormone products. And of course, the immune system is really important here. So this is actually a really interesting link with MHC. So remember, the role of your MHC genes are to help your body recognize and defend against pathogens. So it's defending against anything that's not your own body. Your MHC genes can determine which bacteria your body decides to tolerate, which symbiotic bacteria it decides to let live on your body. And that is the way we think that MHC information is actually transmitted through smells, because it regulates the bacteria you carry, and then those bacteria are producing odors. Okay, so if you found that interesting and want to learn more, I published this little book last year, The Secret Perfume of Birds, published by Johns Hopkins University Press. You can find it online, either directly through the publisher or through any online bookstore. Here's my email address and my Twitter handle. I always love hearing from people. And if there's time, I'd be happy to answer questions.
Wow. That's so fascinating. I don't know how you can keep track of it all and even say all of that in a kind of linear, interesting way. So thank you so much. So let's see who's got questions here.
We have a couple.
In Q&A, somebody's asking, what time of year were your Y maze studies done? Were the birds in breeding season or their status? ?
Yes, that's correct. We only worked with birds who were in breeding condition. It wasn't always on the same calendar time. You can actually manipulate that in captive birds by changing the timing of the lighting. So you can artificially advance their breeding status by turning on the lights longer and longer every day and giving them artificial daylight. So yeah, we have not studied this in non-breeding birds yet.
And Maureen Dwyer asked, does the sense of smell decline as the bird ages?
That's a great question, and I don't think anyone has studied that yet. I would expect that seabirds would be a really great study subject for this because they tend to be longer lived than songbirds. And we do know that senescence, so the loss of certain biological things does happen in birds the same as it does in other animals. Yeah, that would be a good thing to study. I don't know the answer.
So Evelyn McQueen on the chat says, great presentation and great information. Enjoyed it very much.
Thank you.
And Kay Prophet says, very interesting. Thank you. So are there any other questions? I actually have an observation and would be interested in your thoughts about it.
Sure.
If not. I'm struck when you're talking. I love this whole topic and your specific research. I ended up I live with a dog that's part coyote.
Oh.
And he has a noticeable spot, literally a black dark spot, sort of circular on the base of his tail, which reminds me so similar of the preening gland location on birds. And when I lived in Manhattan, somebody came running out of a restaurant and said, do you know that what that is on your dog? She said, I raise wolves, and coyotes and wolves so interbreed, or anyway, that the black spot on my dog is part coyote, which is actually a wild dog, it's a coyote. So that marks the members of its group. So I thought, well, it must be underneath, but actually it seems like it's, I don't know how to pronounce that, the gland, but it's like so similar because it's right at the base of the tail, and
That's interesting.
They preen all the time. You know, but I do know that gland marks their kin, or maybe it's their running buddies. We have, you know, that marks their group versus another group. Maybe, oh, that's not our group. We can fight them, or we'll, you know, we won't share that deer, or whatever it is. I don't know what your thoughts are.
Yeah, I mean, I don't know much about coyote or wolf anatomy, but I know that a lot of mammals, including canids, have a lot of different scent glands. The ones I know about the best are actually hyenas, because my colleague Kevin Tice, that was his main study animal, and they have these scent pouches. I think they're called anal sacs. So they're in that tail area, and they have a paste in there, and that's full of fermentative bacteria. And the way that they share bacteria is that they'll scent mark on the same places, right? And so like a young hyena will scent mark on top of where another hyena has scent marked. And that's actually how bacteria gets into that gland. And so that's how the group members actually all have the same group specific scent. They're sharing bacteria. And there's other examples of this, like, I think, is it beavers? There's some fun studies on them. And they also have some very productive glands. And they groom each other, and it's thought that that's how they're sharing bacteria through that. And yeah, that is quite possible that that's what's happening with your dog there. Very cool.
This may be new information for people that own domestic dogs and fire hydrants. What are they revealing, or what? You could do studies of domestic dogs and fire hydrants.
Yes, you could.
In any case. Let me just see if there are any more questions.
There is.
Yes. Marla Robayo asks, do some plants, trees smell? do some plants and tree smells attract some birds?
Yeah, I didn't talk much about feeding behavior today because it hasn't been my own area of research, but there's definitely evidence that birds are attracted to the scent of the foods that they eat, and that they can use scent to decide which trees to nest near, whether they're going to be fruiting that year, for example. They also, another study that I love is looking at blue tits, which are European bird species. They actually use aromatic herbs that they put in their nest boxing species, and they'll collect herbs and put them in there. And the only function of those herbs is to actually protect the nest from ectoparasites. It keeps bugs off. But they can tell when these herbs have lost their sense of smell, and that it's time to, or not their sense of smell, they're lost their smell, and they can detect that it's time to replace them, which very strongly suggests that, yeah, they're using smell. So they definitely do pay attention to plant odors. Yeah.
Carol Brown asks, do male emperor penguins use smell to find their mates? Do you know anything about that?
Yeah, there's been a few studies looking at penguins, and I think just in captivity in zoos, but they definitely have the same kind of things going on. They have these odors, they're able to detect them. And yeah, emperor penguins, I mean, you see these amazing huge groups of males, and then their females come back and they hand over the egg and they know exactly which one it is. I'm certain that smell is involved in that. Yeah.
So let's see if there's any more questions. What is the significance of the hoopoe to your book or research? I guess that's the bird, pardon me. I know that's the bird on the cover,
Yes.
and I think I'm pronouncing it correctly.
Yeah, so I wanted to put Juncos on the cover for obvious reasons. And, you know, I learned a lot about the publishing process, including you don't always get say over everything. But the publishers thought Juncos looked too boring to put on the cover. And the first iteration of the cover had hummingbirds on it, beautiful hummingbirds. But I said, hummingbirds don't even appear in my book. I don't even say the word hummingbird. We've got to use a bird that's in the book. And I didn't talk about hoopoes at all today, but some of the most important foundational studies looking at preen oil and bacteria in preen oil are all on European hoopoes. So there's some really cool studies looking at these birds. They're fascinating. So their preen oil starts out kind of normal, you know, and it's white and waxy. And then when the females go into breeding condition, it becomes very dark colored and very stinky to, you know, to the human sense of smell. And they actually then take their preen oil and they put it on the surface of their eggs. And it turns out what's happening is when their preen oil gets dark like that, they have tons of bacteria in it, mostly Enterococcus bacteria. And those bacteria produce antimicrobial compounds that help protect the eggs from other bacteria. And so it helps coat the eggs and keep helps improve the survival of those chicks. So very cool birds, beautiful birds. And yeah, they approved putting them on the cover because they're pretty.
So Claire Herzog is asking or commenting, I always heard that dragonflies have no sense of smell, but outstanding vision. Just a little Google search and see that the thoughts are changing. Do you know any scientists studying scent in dragonflies?
I do not. That's fascinating though. Yeah, I did know a scientist studying something in damselflies, but I don't think it was scent. I don't know. I'll have to find out. That's very interesting. Thank you for sharing.
Linda Corwin comments, wonderful presentation. I'm currently reading Slow Birding by Joan Strassman, who references your studies. It's great to hear more details on her chapter on juncos.
Oh, that's great. I have her book. I haven't read it yet. I know Joan. I'm very excited to read her book.
Cool. So I don't see any more questions. One final question is, so if you're in COLDEX studying the oldest ice exploration now in Oregon, What are you doing these days? It's still about birds? That's my final question.
That's a great question. Yeah, so one of the reasons I'm here is because science is funded in funny ways. So I am not a tenure track professor. I've never had my own lab. At Michigan State, I was the managing director of the Beacon Center, and I was able to continue doing my own research through collaborations. But these centers, their funding only lasts for 10 years. And so as time wound down, I didn't really have a job anymore at Michigan State. And this new center here, they were looking for a managing director, and I had all the skills of managing this type of center. And I actually really enjoy research administration. And so I applied, and I'm also completely fascinated by Antarctica. So that was kind of a nice coincidence. And so I'm here now. I'm not doing any bird research at the moment. I'm also not doing ice research. I'm the manager here. But I did actually just go to Antarctica on vacation. Total coincidence. I'd planned that 2 years ago, and I spent a lot of time looking at penguins. I do hope to someday do some work with this center. I think they're doing very important work understanding the history of our climate, which is preserved in the ice cores, and understanding what the climate was like millions of years ago when it was actually much warmer than it is now. So that could be very helpful for us to learn about.
No
But,
kidding.
yeah.
And Antarctica's an amazing, it looks like it was very tropical.
Mhm.
And really, just even, I don't know if you're going to look at that, but maybe we could schedule you several years down the road and have you talk about what you're finding in the ice cores in Antarctica. We haven't had anyone talk about that yet. Jean, did you want to ask something?
No, I just want to say it was an absolutely wonderful, wonderful presentation. And I'm sorry you're not out there actually doing research.
Yeah.
But what I suspect is that you're guiding researchers in your position as a managing director of that. So I hope you do, and I hope you are, and maybe one day you'll get back into it. So.
Oh yeah, I don't think I'm done yet.
Okay,
That's so good.
Yeah.
I second what Jean is saying. It's so nice to have you talk and share your technical perspective in a way that enlightens us all, because you're just it's like you're speaking a foreign language in a way that we can understand it and appreciate not only your results, but you're a very good teacher of it. So I'm so grateful for your time with us. Someone else is saying, great talk, fascinating, thank you.
Well, thank you. I'm really, really pleased to
So
hear
go
your
get her,
feedback,
go
and I
get
hope you
her
enjoyed
book.
it.
That's the way we can thank her.
Yeah, right.
Yeah, that would be great.
Danielle.
Thank you.
Thank you so
Thank you,
much.
everybody, for joining us tonight. What a wonderful, wonderful program.
Yep.
So just before we sign off, we're going to have a local speaker, Sarasota native Kylie Wilson, talking to us about protecting local Sarasota nesting shorebirds on March 13th. And boy, is that an important topic, and God bless her for being out there, keeping all the weirdos from running over nests. And we really appreciate everything she's doing, and we need to give her lots of support. So Kylie's been working on the shorebird monitoring and stewardship with Audubon, Florida since March 2018. Her job includes monitoring our local beaches for imperiled beach nesting species such as snowy plovers, black skimmers, and least terns. And she also coordinates volunteers. So please, everybody volunteer soon to educate beachgoers about these species and preventing disturbances. So please join us again next month. And thank you so much for tonight.
Thank you all.
Good night, everybody.
Thank you.
Good night.
Good night.